quarter, and I think the next one's on January 7th. We begin the presentations again, and it's you know, it's a lot of fun to have Lenny Tender here with us. Lenny, actually, we just chit-chatted. We overlapped at MIT. He was an undergrad with Mark Wrighton, that's a very famous electrochemist at the time, and then went on to all sorts of other things. So he got his bachelor's there, and worked with, and then got his PhD at the University of North Carolina, Chapel Hill, with Royce Murray, and then postdoc at Berkeley. So he's now at the Naval Research Labs in the Washington area, and he works in the area of bio-electrochemistry, an emerging area that is quite fascinating because it incorporates living systems with devices and materials and all sorts of finishes that are very interesting. And so it's also worth noting that Lenny has received several awards, including the 2014 ISMET Service Award. He's also an Associate Editor of Frontiers in Energy Research. And he, in the year 2011, obtained the Arthur S. Fleming Award for Exceptional Federal Service. And this is an award that is there prepared by the government to award outstanding federal employees. And his bio for that award reads that he's the Department of Defense's leading science and technology expert in the field of bioelectrochemical systems, processes in which microorganisms are used to catalyze electrode reactions. And I think that's going to be more or less the theme of his presentation today. So in the last few minutes that you have for introduction, you can. It's good to have you.
Oh, thank you. Thank you. It's such a pleasure to be here. So concerning that first award, the ISMET Award, that's the International Society for Microbial Electrochemical Technologies. It helps if you co-found the organization. And then so that's a little advice I see a lot of young people here. It's a good way to get an award is to create your own organization, which we did. And concerning the federal service, yes, I'm a very proud federal employee. Does anyone know if we passed the budget today? I've been traveling.
New York Times says there's a deal.
Okay, lights are still on. Great. All right, so I can fly back home. Yes. Okay, so this is one of my favorite images I'm going to start here with. This is from Daniel Bond's group at the University of Minnesota. And this is a Geobacter sulfur-reducing biofilm grown on a graphite electrode. And so if this works here, this sort of canyon here is actually a crack or a crevice in the biofilm, and that's the electrode surface. And each one of these little macaroni-looking things is an individual organism. They're on the scale of about a micron long, maybe half a micron wide. And the really cool thing is that these guys are living off of this electrode. They're respiring the electrode. What they're doing is they're oxidizing acetate, which is a fuel. It's a carbon source, and it's also an electron source. Microbes need both. And they utilize some of the carbon to build up their body mass. What they don't use for body mass gets kicked out as carbon dioxide, the oxidized form of the carbon. And, but the electrons that they get, I've come to think of microbes and really all organisms as little electronic machines. The electrons enter the cells with some potential energy. The cells extract potential energy, and they have to get rid of the electrons. And that's just basic respiration. And the electrons are flowing through the biofilm from the individual cells onto the electrode and out. And that's really fascinating. My training is in electrochemistry. And you can create this kind of a biofilm in a very nice, clean electrochemical reactor. You inoculate it with cells and grow it up. And essentially, the microbes are catalyzing an electrode reaction here. They're catalyzing the electrode oxidation of acetate, pulling the electrons out of acetate. From their perspective, they're respiring because they're taking a little bit of the energy of the electrons from acetate, and that's how they're making their living. But it's a fascinating area. And what's really fascinating is just how, like a microbe that's up here, maybe 20, 30 microns off of the electrode surface with all this stuff between it and the electrode. How is it pumping its electrons down onto the electrode surface? And that's a really fascinating area that many of us are engaged in studying. A big part of it has to do with the concept of metal-reducing bacteria. So this is another image that I stole from the lovely lab at UMass Amherst. And these are Geobacter again, and this time this is more of the natural situation. Geobacter, this is Geobacter metallo-reducens, is a pervasive, Geobacter species are pervasive in what's called the subsurface environments and sediments and soils and marine environments. And they are iron-reducing bacteria. So a lot of iron is found in these environments in insoluble forms. So this is ferrihydrate. It's iron in the oxidized form, and it's insoluble. And this is how it may be found in sediments and soils. And the microorganisms are actually latching onto it, and they're pushing their electrons onto this insoluble iron, which is acting like an electron acceptor. And that in itself is very fascinating because the dominant paradigm in microbiology is that you have a soluble electron acceptor like oxygen, and it permeates the cell membrane, gets inside the cell, gets reduced, and then carries the electrons out. And what these guys are doing are actually pushing the electrons outside of the cell and onto the iron surface, which is really sort of a very interesting process. And the biofilm that I showed you in the previous slide is sort of taking that one step further. In sort of an unnatural environment on an electrode, you can actually get the cells to stack up very on top of each other and form thick biofilms. The reason I think, and it hasn't been really definitively demonstrated, but I think is that as you start reducing this iron mineral, it becomes soluble. So there's a finite capacity for this to accept electrons. The electrode in the previous slide that I showed you, if it's attached to a potentiostat or some electrical circuit, can accept electrons forever and never becomes soluble. And so that allows the cells to proliferate and grow and grow and grow into thick films. Anyway, so to sort of take this idea of metal-reducing bacteria one step further, one of the other most highly studied metal-reducing bacteria is Shewanella. And the key thing I wanted just to express to you here is that these organisms, Geobacter, Shewanella, one of the defining properties, of course, is they have two membranes, an inner membrane and an outer membrane, periplasm in between. The cytoplasm would be here, and this would be outside of the cell. And in order to get the electrons from inside the cell, where the reduction or the oxidation of the organic matter occurs, and to get it out of the cell requires an electron transport chain or process. And in Shewanella, it's the mechanism is much more highly resolved than it is in Geobacter, although neither mechanism is definitive. And just the key thing I want to point out too is that the key features is that you have multi-heme cytochromes. Hemes, of course, are iron complexes, single atom iron complexes, and pervasive in biology. And electrons can hop onto the iron, and the atom can be oxidized or reduced. And so here you have molecules that will have multiple hemes, 10 hemes, 12 hemes, and they kind of act like little conductors, and electrons can hop from iron to iron in discrete steps. The closest analogy I can think of from my background in electrochemistry is a redox polymer, if you're familiar with those. And that's very fascinating. We usually think of electron transport in biology, maybe on the distance of a single molecule, but to recognize that you can get, 3 different types of cytochromes here and as many as 21 or 27 hemes involved in electron transport. In this case here, coming out of the periplasm through the outer membrane up to reducing an iron oxide mineral here. But also there's other outer membrane cytochromes that the electrons might go one way or another and reduce other intermediates and whatnot. So that's a pretty fascinating concept. I just want you to keep that in the back of your mind for a few minutes. We'll come back to that. I want to touch upon I showed you that first slide, and I want to touch upon what I think is the state of the art in terms of using microorganisms that are able to push electrons onto insoluble oxidants in the environment, sort of using them to push electrons onto electrodes and making them into catalysts. It turns out that there are organisms that can work in the opposite direction. They can use electrons that are delivered from an electrode and drive a reduction reaction. So in the first case, an anode is an electrode that accepts electrons, and a cathode is an electrode that delivers electrons. And which is really fascinating to me as an electrochemist is that you can essentially inoculate an electrochemical reactor with anything that has microorganisms in it. Wastewater is typical, sewage, seawater, marine sediment, and there's a diversity of organisms in there that can either reduce an electrode or oxidize an electrode. And you can pull them out with very simple enrichment techniques. I mean, if you just dump a little bit of wastewater into an electrochemical reactor and poise a graphite anode at a sufficiently positive potential, you will form a biofilm. It'll be enriched in a subset of organisms that were in the inoculum that you used, most likely dominated by Geobacter, and you'll start generating current as the cells are turning over acetate organic matter and generating carbon dioxide. So I've been around people for 30 years who are trying to build exquisite electrode catalysts, and here you can just squirt some wastewater into your reactor, and it's quite beautiful. The applications here are fairly limited at the anode side. You can oxidize organic matter, and the two principal reasons you'd want to do that is to generate power. So if your organic matter is a fuel, and you could have a sugar battery, for example. The other thing is you can use it to oxidize organic waste when you just want to clear out the waste. And so, for example, wastewater treatment. A big part of treating wastewater is oxidizing the organic waste that's in wastewater. And there's a huge amount of energy that is consumed in doing that. Typically, air is blown into wastewater to promote aerobic digestion of wastewater. Here you can actually maybe cultivate electrodes with biofilms and have them just digest the organic matter directly. You might have to push in, or you have to pull out electrons, and you might have to attach this to a circuit that ultimately consumes power, but it may be solar power you're consuming, and it may be more energy efficient. Those are really the two things you can do with an anode. They're still fascinating, though. To me, cathodes are much more exciting. There's a lot more diversity on the cathode side. So we just published a paper where we collected some sediment from a wastewater treatment plant. We enriched in a culture on the bench for microbes within that sample that would oxidize iron compounds and reduce nitrates, and then took the result of that culture and put it into an electrochemical reactor, and lo and behold, we had a biofilm that could take nitrate to nitrogen. And that's another important component of wastewater treatment. But another thing you can do is cleave carbon-chlorine bonds. So there are microbes that do that in the environment, and they need electrons to do that, and you can form a biofilm on an electrode that would do that. We've just submitted yesterday a third paper on a biocathode that we call MCL now, that essentially came from seawater, where we inoculated an electrochemical reactor with seawater, which has organisms in it, and over a course of different cultivations was able to generate a biofilm. That it primarily does is it reduces oxygen to water. That's how the cells in the biofilm live. And that's a pretty mundane thing. But they take some of the electrons they get off of the cathode and some of the energy they get from that process, and they use it to reduce or fix carbon dioxide for biomass to grow. And that's really the exciting thing. The biofilm is comprised of a consortium of organisms, maybe 6, 8 different organisms. We can't separate it. We can't break it down any more than that. And it looks like there may be 2 or 3 keystone organisms that are involved in this oxygen reduction to reaction and this fixing of carbon. And what that means is there may be one organism that is able to take carbon dioxide and generate it into a form of reduced carbon biomass and then share it with the rest of the consortium. And that to us is very exciting. So what we'd like to do is be able to sort of siphon that off. And this is a concept that's called electrosynthesis. You're actually taking carbon dioxide and making organic matter out of it, reduced carbon, and it could be a biofuel precursor, for example. And that's a huge area of research in this field. So I want to, since there's a lot of students here, I thought I would tell you how I got into this, because I find it interesting for myself, at least, and it's not at all rational in any means. But back in 1998, when I was finishing my postdoc and trying to figure out what I could sink my teeth into, I learned about a phenomenon that is referred to as the EH gradient, which is widely known in marine geochemical circles. And essentially what it is if you take an insulated microelectrode, where the only part that's exposed is the tip, and if you stepwise insert it into marine sediment, the deeper you go, you will measure a more reducing potential. So if you measure the potential up in water, it's going to be about 0.6 volts versus hydrogen. And as you go deeper on the order of millimeters to centimeters, the electrode tip will start to swing negative on the order of about 0.8 volts or so. And we had this very sort of, at the time, my collaborator, Claire Reimers, who was a professor at Rutgers University, we thought, hey, you have a voltage gradient. Maybe we can generate power. Maybe it'd be like a battery. You have an electrode up here in the water, an electrode down here in the mud. We can just connect them and see if we can generate power. Let me tell you a little bit about where that gradient comes from. These equations represent the metabolism of organic matter by microbes. And it's a general scheme as a function of depth. And the key observation is that marine sediment is really a matrix that has a lot of organic matter in it. And that's represented by this equation here. And essentially, glucose that derives from anything that has lived in the overlying water, like phytoplankton, will eventually settle down under the influence of gravity and accumulate onto the sediment surface. And that's what this is. Microbes will live off of it, oxidize it, and they need an oxidant. And most of the oxidant is supplied from diffusion or advection from overlying water. And so not all oxidants are equal. Oxygen is the most potent oxidant. And so microbes at the top of the sediment surface will preferentially consume oxygen and deplete it. So you'll have a zone of aerobic breakdown or metabolism of organic matter giving you, each cell per equivalent of organic matter the most energy return. And quickly the sediment becomes depleted in oxygen, and typically below the first few millimeters or so, you're essentially anoxic. And then they start going through a sequence of less potent oxidants, because if you're a microbe up here, you're not going to use iron because you can get out a lot more energy per equivalent of organic matter. So you get this sort of stratification of the, people call it redox chemistry, metabolism. But the end result is, if you are a microbe and you're using a pretty weak oxidant, like sulfate, you're generating, when you reduce it, a pretty strong reductant, which is sulfide. And so when you start stepwise inserting that electrode into the sediment, what you're really measuring is the accumulation of reductants deeper and deeper in the sediment. And so that was how this is traditionally explained, although this model has come under attack in the last year or two. But nonetheless, we had postulated that, well, if we just create a battery out of the marine water interface, we put an electrode in sediment, an electrode in overlying water, this will act like an anode. It's going to oxidize whatever reductants the microbes have accumulated down there. And this guy here is in the overlying water. There's usually a lot of oxygen, so we have an oxygen cathode. One of the sweet things about this device is that You don't need a proton exchange membrane or any other barrier to keep oxygen from getting into sediment because you have that top layer of aerobes that are going to deplete oxygen for you. And so you don't have to put any sort of membrane across here. So we had postulated that we can generate a battery from the marine sediment water interface. You know, why would we want to do that? Well, I work for the Navy. And the Navy puts out a lot of sensors into the marine environment. They're battery powered. That means they have a very limited lifetime. And this is a way in which we could perpetually power sensors in the marine environment, get away from the depletion of battery power. So it was a harebrained idea, but we did it, and we did the first proof of concept in shoebox-sized aquaria in the laboratory. Just collected some sediments, put them on the bench top. And even though, you know, you go out there with a shovel and you homogenize the sediment, it gets all mixed up, you put it on the bench top, pretty quickly that gradient reestablishes, and you can measure the voltage gradient within a matter of a day or two. It reestablishes. We put a couple of electrodes across the interface, and we characterize the voltage current relationship. Each one of these dots, it represents a different resistor placed or resistance placed between the two electrodes, and we watch current flow. And as you reduce the resistance, current would flow. On this axis, we'll look at the blue curve here. And if you measure voltage, the voltage would drop just like any other electrochemical power supply. And this voltage current relationship is just textbook for a fuel cell. It happens to be mass transport limited fuel cell. That's why when you take the product of current and voltage, you get which is power, you get a semicircle curve like this that rolls over. And it's just a textbook. And what it says is that we could generate power in this one environment. This was from sediment in New Jersey, actually two places in New Jersey, very different sediments. This had a lot of sulfide in it. This is a sulfide concentration profile versus depth. This is a different sediment. Not all sediments have sulfides. There's variability in the redox chemistry, but the phenomenon of the gradient is pretty universal. And we got a different, the same type of qualitatively, the same type of curve. And so we could sit here all day at this resistor and pull current at this level and this voltage and get this amount of power up here, indicate on this axis. So we could generate power and it was mass transport limited. So that was pretty cool. So then we did it in the real environment. And this is the Naval Research Laboratory in Washington, D.C. We sit right on the Potomac River. If you've ever been to Washington, we're right across the river from the National Airport. And we have a nice pier that goes out into the river. And so this is a very sophisticated design here. We have a milk crate, some big slabs of graphite, and we just zip tied them on, one on top and one on the bottom. And this constitutes our anode. There's electrical connection there, and this is the cathode. And we just went into the river and pushed the milk crates down, and so the mud level was about here. And that was our fuel cell, and it worked. It worked beautifully, and it had a little weather monitoring system on board and a little radio transmitter. And so every 5 minutes, I would get a little signal sent to my office with the air temperature, the water temperature, the air pressure, and the relative humidity. It took a little bit of finagling to pull this off. As I mentioned to you earlier, the voltage drop across the interface is about 0.8 volts, which is pretty low. And when you operate a fuel cell, a mass transport limited fuel cell, you typically operate at half of its open circuit voltage. So we're operating at about 0.3 volts. And there's not a lot you can buy at Radio Shack that you can power at 0.3 volts. So it took a little time to develop some DC to DC step-up capability. So essentially there's a little package in here that upconverts the 0.35 volts to 6 volts to charge a capacitor, and the capacitor runs the monitoring system, which had a very, very little draw on it. The big draw was the RF transmission every 5 minutes. But we did this. This ran great for 7 months. The river froze over that winter, and then it broke into pieces big enough to start flowing downriver, and this got dragged downriver by an ice flow. The lesson is you put your name and phone number on your oceanographic instruments, because I got a call from the Coast Guard and I retrieved it. but it was a nice little demonstration. So a big chunk of my time now, and that picture that I've seen plastered all over campus of me in the wetsuit, is that we are now implementing this on very large scale moorings. Typical mooring is 2 meters in diameter. Looks like a big Hershey kiss. It weighs about 900 kilograms. And that's where an oceanographic sensor will sit in. And we put the anodes on the underside of the mooring, and then we throw them off of ships. And they're supposed to go down, and the anode's supposed to bury into the mud, and we have a cathode on top. And we have a big indoor facility with a big 18-foot diameter tank that's full of sediment and seawater. So we can optimize the designs indoors under controlled conditions before we go out to sea, because it's very expensive to do iterative design development in the marine environment. Ship time's very expensive, and there's a whole host of logistical problems. But indoors, I can do 10 experiments in a week, so it's a very good situation. As I said, we're mass transport limited. We're mass transport limited at the anode, presumably by the rate at which organic matter in the sediment. Oh, actually, I'm getting ahead of myself. I'm telling you about what we think is happening. Let me come back to that. So how's it working? That's the key question, you know. So one of the first experiments that we did is we put 2 of the fuel cells side by side in the marine environment. And one of them, we actually connected the leads across the load, and we were generating current. The other one, we left at open circuit, so that the electrons had no place to go from the anode, and they weren't going to go through any circuit to the cathode. And we let them sit out there for, you know, I think 9 months. We brought them back into the laboratory. They each had the anodes, the ones in the electrodes and the muds, both had biofilms on them. And we scraped them. And the key observation is that even though they had biofilms, the one that was drawing current had an enrichment in a certain class of organisms that contains a lot of the metal-reducing bacteria, the Geobacter species I mentioned earlier. So this is a comparative community analysis, and the one that was generating current had a much higher abundance of the metal-reducing or iron-reducing bacteria. And so that was a clue to us that those bacteria were involved in the anode reaction. And so then if you go back to the laboratory and you do an experiment like in that very first slide that I showed you, if you just take a pure culture metal-reducing organisms like desulfur-ammonium acetooxidants, inoculate it into your electrochemical reactor, and then inject a very, very simple organic fuel source like acetate, and provide it no other electron acceptor other than the electrode that's poised at a positive potential, all of a sudden, whoosh, you get current flow. And you'll form a biofilm. The cells will stick to the electrode and start turning over the acetate, generating the electrons, and that's where the current comes from. And so the model that we have now of that sediment fuel cell is that we get spontaneous colonization of the anode of these metal-reducing bacteria that are drawing acetate out of the sediment. Acetate is not really present in a high concentration. It's turned over through fermentation processes from glucose by other organisms. So you can think of the sediment as a big bioreactor with a lot of fermentation going on. You form a catalytic biofilm on the anode that's drawing off the product of that, getting the electrons, and then there's another biofilm that's formed spontaneously on the cathode that's driving oxygen reduction. The thing about biofilms is that they're incredibly durable. They last, I mean, you can't, it's nearly impossible in the natural environment to kill a biofilm. So once this thing sets up, and it sets up spontaneously, we've had systems that have run for cathodes in my labs for 5 years, and they just keep on running and running and running and running. All right. So, and the other thing I'll just tell you is since we're mass transport limited at the anode, meaning that either the turnover of glucose or acetate, something down here is limiting, that means all these moorings that I'm doing, we have a lot of surface area of anode that we're jamming into the sediment to boost up the net rate of the electrode reaction. That's sort of the general solution of a mass transport limited electrode reaction, is you try to get more electrode into your reactor, as long as you don't get overlaps of the depletion zones around adjacent areas. So now, in the last, say, 15 minutes or so, let's talk a little bit more about the harder science here. As I'm very intrigued about, this is a cross-sectional image of a Geobacter sulfur reducens biofilm, and I'm very intrigued about exactly, you know, how does this microorganism that's here, that could be, you know, 20 microns away from the electrode surface, how is it living off of this electrode? How is it getting its electrons to the electrode surface? As an electrochemist, to me, that's a really just interesting fundamental question. So that's the question that we've been studying for the last 5 or 6 years, maybe even longer. So we're proponents of a model in which the transport process is essentially a hopping model, and it's an extension of the model I showed you on the second slide of the Schuonella electron transport that's inside the cell to get to the outer surface. Here we think it's an extension of that to the outer extracellular domain, to the materials that cells secrete on the outside, that's sort of the mortar that keeps the biofilm together. And one of the defining properties of Geobacter, for example, is that the genome indicates that there should be at least 100 different outer membrane cytochromes. And when you grow up a Geobacter biofilm on electrode, it looks pink from all the iron. So there's a lot of cytochromes in the extracellular domain. So we're a proponent of a hopping mechanism. The cool thing about that is we're talking about electrons hopping possibly from hemes or through hemes within a cytochrome to an adjacent cytochrome, maybe a web, a network, over distances that are tens of microns, multiple cell lengths as opposed to just, across a membrane. So we think that's a pretty cool thing that could be happening. And so we're sort of showing it here schematically. The gray ovals are cells. Cells will have on the outer surface of the cells, outer membrane cytochromes. There'll be cytochromes that are in the extracellular materials. And then a very complicating issue is that cells will secrete filaments called pili. And cytochromes some cytochromes align on pili, but also pili are associated with the actual secretion of some of the cytochromes out of the cells. So it's very, very hard to separate pili from cytochromes. And there's another model of electron transport through a biofilm that says that the pili act like organic conductors. But sort of separating the two is very, very difficult. So I'll leave it at that. We're a proponent of the hopping model as opposed to the intrinsic coherent conduction model of the pili. Here's some pili. Again, this is other images also from the lovely laboratory in Ruggiero lab at Michigan State University. Here, these are pili from the cell in culture. These are gold labeled antibodies for outer membrane cytochrome. I think it's S that's aligned on the pili here. And this is showing, I believe, ferrihydrate that's associated with pili, and just showing that the cells are using these pili to maybe ensnare the metals, and the cells are actually dumping electrons onto these metals. But are they doing it themselves, or are the cytochromes involved in the transport process? That's really the blood sport right now in this field, is trying to understand how that's happening. A little bit more about pili. Pili are made from pili proteins that assemble into these filaments, and they have a helical core. And so the proponents of the intrinsic conductivity of pili believe that the core, the helical core, becomes sort of an organic conductor, which I think many people at this institution are very familiar about. And I showed you a little bit about the cytochromes earlier, but here's a crystal structure, or an image based on a crystal structure, just showing that you have iron centers. And this is not even an outer membrane cytochrome, but just as a representative, the cytochromes, multi-heme cytochromes, exist in these systems to act as a transport pathway for electrons. And we believe that the same process is happening outside the cells and inside the cells. So there's really three tools, three hammers that we have to try to get some insight on the mechanism. First of all, I'm an electrochemist. And so what that means is I can adjust the potential of the electrode here. And if the electron transfer reaction between the last cytochrome and the electrode is fast, then I can adjust the relative concentrate. I can adjust the concentration of the oxidized form of this versus the reduced form of that. And that's what the Nernst equation tells me, and that's what this is here. So the more oxidizing I make my electrode, these are going to be oxidized. The more reducing I make my electrode, they're going to be reduced. So the electrode potential should influence the oxidation state of these guys, whereas everything else is being influenced by electrons being dumped into this pool by the turnover of acetate from the cells and pushing electrons that way. So in a sort of energy diagram, if I'm going to have a hopping mechanism, I have to have a redox gradient. I have to have a difference in potential to drive electrons from one site to the next site to the next site and onto the electrode surface. And I can create that from adjusting the electrode potential. So if my electrode potential is fairly positive with respect to the formable potential of those cytochromes or those heme molecules, then the more oxidizing, they're going to be more oxidized close to the electrode surface, and that means is that their essential redox potential is going to be more positive. They're going to be more reduced closer to the microbe that's feeding the electrons into the cytochrome pool, and that's going to make its effective redox potential more positive, and I'm going to have a gradient. So another way of saying that is, in order to have a gradient, an energy gradient to drive electron transport from a cell turning over acetate generating electrons to an electrode surface, I have to have a gradient and oxidation state of the molecules through which the electrons are hopping. And I can influence that with the electrode potential, because the electrode potential is setting the boundary condition of that last site. So if I make the electrode potential, for example, more negative, well then more negative than the formal potential, I can even push charge into the cytochromes from the electrode surface and shut down metabolism. So I can control that. Taking that one step further, if we're able to look into a biofilm that's catalytically active, the farther away I get from the electrode surface, the more reducing or the less oxidized the cytochrome should be. And we can actually measure that. Cytochromes are really Raman active. And so we've published a few papers recently on doing resonance Raman spectroscopy of Geobacter biofilms on, in this case, gold electrodes. And not only are they very Raman active, but you can actually detect changes in their oxidation state. So this is a confocal measurement. We're actually going into the electrode from the top of the biofilm into the biofilm down to the electrode surface. And the deeper we go into the biofilm, the more oxidizing the cytochromes are. That's an ex situ measurement, though. We were scooped. A group had done the same measurement in situ when the biofilm is actively turning over acetate and adjusting the electrode potential. So at a very negative potential, all the cytochromes are reduced. And as they adjust the electrode potential, more positive indicate on this axis here, the closer you get to the electrode surface, the fraction that are oxidized increases. And so you have a gradient that was measured there. The other thing is that electron hopping is very analogous to diffusion. In this case here, the electron is essentially diffusing through a pool of redox sites, hopping, doing one step at a time. And if you have a pool of redox sites and you're an electron, any given electron has an equal probability of going one way or the other. It's just the fact that you have more electrons that are being generated inside the biofilm, or let me put it another way, you're generating electrons in the biofilm and you're extracting them from the electrode, that you have an imbalance. You have more higher concentration of electrons in the biofilm farther from the electrode surface, and that drives diffusion, as if the electrons were a soluble molecule that are diffusing, that are able to diffuse. So the cyclic voltammetry is very clear evidence of a diffusive process. If you remember in that slide I showed you the energy level, maybe I'll go back, where I can, here I adjusted the potential very positive, and I'm able to suck electrons out. And then here I adjust the potential very negative. And what I didn't show here is I can actually drive electrons into the pool and reduce these. And then if I put the electrode potential back positive, I can actually pull electrons out and reestablish the gradient. You can see that electrochemically. So at a very slow scan rate, where I'm changing the electrode potential in a very slow manner, each point along the curve is considered steady state, and I can't capture the rush of charge going in and out of the biofilm as I'm changing the potential. But if I speed up the rate at which I change the electrode potential here, if I start sweeping the potential negative, I shut off the catalytic current. At the same time, I'm pushing charge into the biofilm from the electrode to reduce the charge carriers of cytochromes. And that gives me a transient current peak. And then when I reverse the scan and I come back, I can suck electrons out from the electrode at the same time as the metabolism is restarting. So the shape of this curve is very classic for a diffusive process, as if the electron carriers are diffusing physically. Instead, they're fixed in the biofilm. The electrons are hopping. At least that's our interpretation. The shape of the curve, and if you were to do what's the classic analysis is to look at the scan rate dependency of the peak current, and it goes with the peak current goes linearly with the square root of scan rate, and that's a classic diffusive signature. So the last bit I want to show you is in the last few years, we have been working on interdigitated microelectrode arrays, and these are electrode bands that are fabricated onto, they're typically gold, micron dimensioned, maybe 10, 15 micron gold bands. This is the cross section with 10 to 15 micron wide gaps between them. And you know, they're set up like a comb effect so that every other one terminates on one side and every other one terminates on the other side, and they don't touch. So essentially I have two electrodes that are like combs that are pressed into each other, but the fingers never touch. And the appeal of that is if you trace out the gap between the electrodes, it's sort of one continuous gap. So really what I have is two electrodes and one long gap between them. And what we do is we form a biofilm over the entire array, and then we put a bias between the electrodes. And it's the same thing as if I had two really, really long electrodes, and I'm going to put a bias between them and then measure the current from one electrode through the biofilm to the other. This allows me to get about 45 centimeters worth of gap in something that is, you know, a few millimeters on edge. So I can actually get it into my electrochemical reactor. So it's a very good, and of course the longer the bands are and the longer the gap is that you're growing your material over, the more current you can get and the better signal to noise is in the measurements. This was exciting for me because IDAs were used heavily in the Wrighton lab when I was an undergraduate, and I never appreciated it. So I'm citing these papers back from the early 80s in this field from people that I knew, and I still knew. And then when I went to graduate school in Chapel Hill for Royce Murray, Royce was working on IDAs also, and I wasn't working on that project either, but you know, I'm citing his papers as well. So this is a Geobacter sulfur reducens biofilm that is grown over the IDA. So essentially it poise all the electrodes, you know, every other, again, it's really two electrodes, and looking at the cross section, this electrode and this electrode are parts of the same electrode. This one is part, and this one are part of the same electrode. If I poise them all in oxidizing potential, biofilm will form over them because they're acting as an electron acceptor for acetate oxidation. And then eventually, if the dimensions of the gap widths are smaller than the typical thicknesses of the biofilm, the individual biofilms will merge and you'll form a conformal biofilm across the entire array. And then you can start poising the electrodes at different potentials and ask yourself, if I put a bias between the electrodes, can I sort of hijack the electron transport process and drive electrons in from one electrode through the biofilm to the other, sort of independent of the acetate metabolism, and just look at the electron transport process. And so in this sort of energy scheme here, you can think of it as here the energy, here's the two electrodes, the relative energy, and the key thing is I'm going to keep this one is poised at a more negative potential than this one, but they're both negative of the formal potential of the theoretical charge carriers, the hemes of the cytochromes in the biofilm. So they're all oxidized here. But if the electrode keeping the same bias between them, if it straddles the formal potential, then I'm going to essentially make the ones that are closer to the electrode that's more positive potential more oxidized, the one over here more reduced, and I can drive electrons in from this side, they'll hop and come out on this side. And the other thing to keep in mind is now if I shift the electrodes even more negative, now they're both negative of the formal potential, and all of the sites are going to be reduced because in the short term electrons will flow into here, and the whole thing will be clogged up with electrons, and there's no way to get them out from this side or from that side. And so the hopping mechanism in a two-electrode measurement like this predicts an intermediate or peak-shaped dependency on the current that you would measure between the electrodes versus the average potential of the electrodes, and it should be centered on the formal potential of the cofactors involved in the transport, and that's exactly what we see. So here we are sweeping on this axis. This is the average potential of the two electrodes in the IDAs. The biofilm was grown across it, and here I'm measuring the current going in one electrode, electrode 1, at the same time the current going out the other electrode. So it's sort of two measurements of the same current. And we get this beautiful textbook peaked current gate potential dependency, which is, again, very consistent with a hopping mechanism in which you have redox cofactors that you're oxidizing and reducing. Here, on this side here, they're all oxidized. There's no charge going through the system. Here, if I was able to extend it further, they'd be all reduced and there's no charge going through the system. In a hopping mechanism, the most conductive region is when you have 50% occupation. 50% of the sites are oxidized, 50% are reduced. And that's in the absence of acetate. So we starve the cell, we got them growing, we form the biofilm, and then we wash out the acetate. And the biofilms are still viable, they're just not metabolically active. We can do the same thing when acetate is present. This becomes a little bit more complicated, and every time I try to explain this, even to people in my group, I get blank stares. But in this model here, not only would I have electrons going from the more negative electrode to the more positive electrode, but if either electrode is sufficiently positive to support metabolism of acetate, I'm going to have electrons coming in to the electrodes due to the turnover of acetate. So the electrodes could both act as anodes, and one can act as a source and one can act as a drain. So you get the superposition of the two currents. And so you get a catalytic current and then superimpose on that the peak currents. And we're able to background subtract out the catalytic current. In other words, say from control measurements, say, okay, let's subtract out the current that's due to the turnover of acetate at both electrodes, and get qualitatively the same peaks as I got in these slides, in this non-turnover condition here. And so what it says is, if the cells are metabolizing acetate, they're not metabolizing acetate, the same process of a transport through the electrodes, through the biofilm is occurring. So the last thing I'll show you is, this is a very, very early biofilm. This is a paper we just came out very recently, very early in growth. And these are the bands, and these are cells that are stained with a dye that allows you to localize the cells. And essentially what happens is you'll get a cell that'll stick onto the electrode surface or the glass surface between the electrodes, and then it'll start proliferating and forming clumps. This is a stain that's specific for sugars, for polysaccharides, which is the predominant component of the extracellular ooze. These cells are secreting all this ooze with the cytochromes and the filaments and other things in them. And it's almost like looking at an egg, a fried egg. You got the cells or the yolk, and then you got all the whites that are extending, beyond it. And so the point is between the electrodes there's not a continuous cluster of cells that could support conductivity, but there are continuous, domains of the extracellular material that are connecting the electrodes together. And lo and behold, we get qualitatively the same result, the same type of gate measurement that I showed you earlier. The signal to noise is much worse because the current is much lower because we have a very, very thin biofilm. Essentially, I'm looking at current going through clusters or through material, between the two electrodes as opposed to the very big biofilm. But I'm still seeing a peak here and the peak associated with it here. And there's other stuff here going on. We're not quite sure what that is. But the take-home lesson is that even in a very, very early state of growth, when the biofilm is really just a discontinuous collection of cells on the surface, but a lot of ooze between them, it's exhibiting conductivity, which is consistent with a hopping mechanism. And it may be that the cells are secreting a lot of cytochromes and whatnot to facilitate electron transport. Anyway, so I'll stop there and just acknowledge my collaborators, Sarah Glavin, who was my former postdoc and now my colleague, Jeff Erickson. Rachel Snyder was a former postdoc, Stanislav Sloy, and Nikolai Lebedev. Nikolai is really great doing the Raman spectroscopy. He's joined my group in the last few years and knocking out a lot of interesting papers, both on the measurements, single cells on electrodes, but also just submitted a paper on the theory of whether or not, pili can be conductive, for example, and funding from Office of Naval Research and NRL-based funds. And I appreciate your attention and be happy to answer any questions.
Questions?
So, about this cytochromes.
Yeah.
Hopping mechanism. Assume that all these electron transfer outside of the cells are done by cytochromes. Have you
Sure. So, yeah, so that's a very good question. Yeah, sure. Back of the envelope calculations we've done, and the answer is that you don't need to have a continuous matrix, a solid matrix of cytochromes. You could have something like 1 or 2% of the total volume of the biofilm be represented as continuous heme domains through which the electrons would hop. And it's the same argument that, for pili, if how conductive a pili is, can a single pili support all the current that a cell generates? And I think in the case of Shulnella, the answer is yes. That's been Mohammed Al-Najjar at USC, I think he's done those calculations. Interestingly, as you probably are aware, one of the recent papers on the Shulnella pili turned out to be that it was a membrane extension with a lot of cytochromes on it, and that's the mechanism of hopping. And I think that was more established. The hopping is much more established for Shulnella. It's still up in the air for Geobacter. But to answer your question again, a very small portion of the biofilm volume would have to be cytochromes to support the current that we're seeing.
So I assume that means that it does not have to be all homogeneous in the biofilm. They just have to be aligned sort of way that they align in the pili, for example.
Sure. So the point, one of the points, the subtle points of that last image here is that when you have a very sparse biofilm, it could be that all the current is conducted by scooting along the cell surfaces on the outer membrane cytochromes. And if you have cell-to-cell contact throughout the biofilm, that could get you from a cell that's here to the electrode surface. I think one of the subtle points here is that you don't have continuous domains of cells here, so the matrix between them must be bridging at least the gap between cells, if not carrying all the current. But your point is absolutely right. It can be totally heterogeneous. And that's important to know because people want to quantify like biofilm conductance. Give a number how conductive is a biofilm. But what about all the void space where the cells are sitting? How do you handle that in your calculation, right? So.
Yeah, I was very curious, like if those cytochromes are more keen to the pili, like for example, the cytochromes prefer to be aggregate around the.
Sure.
What's the driving force for that? Are they, like for the Shulnella's, they produce on the membranes, so they were embedding the membranes. But how those cytochromes?
So I'm an electrochemist, so this is not my comfort zone. But from what I understand, is that in Geobacter, the pili secretion draws cytochromes out with it. So it's part of this cytochrome secretion process. So it may be that they stay associated with them.
Other questions? Because I think you guys.
I'm done.
Okay.
Yes,
We could talk over dinner, I guess.
One of the things Mark Reitman was enamored with was being able to turn reactions on and off.
Yeah.
Electrode potential.
Yeah, yeah.
The first slide you showed and you explained what it showed, I was thinking when I saw it that, wow, you can turn life on and off.
Absolutely.
Changing the
Yeah.
potential of the electrode.
Yeah.
And can you measure like what potential window it will live? Can you kill it?
So that's a very interesting question. So, so I didn't describe this curve, but this is the current voltage curve of metabolism of a Geobacter biofilm, right? Of maybe the biofilm on the first slide. If I shift the potential very positive, those cells are very happy and they're respiring. If I shift the electrode potential very negative, it can no longer accept the electrons. I'm turning it off. Okay. So the paper we just submitted on the other day, what's today? Today's Wednesday. So we submitted it yesterday on our biocathode, MCL. We actually operated it at 2 different potentials, and so it's a cathode, biocathode that's comprised of a consortium of organisms. And when we changed the electrode potential, we changed the rate of the electron, of how the rate of uptake of electrons the biofilm can handle from the electrode. And then we went into it and we asked, okay, which proteins are being turned on and turned off? And so that's an omics. So we have a metaproteomics and metagenomics effort going on, metatranscriptomics also. And so we're trying to get into the nitty-gritty of which processes in the respiratory chain can we manipulate with the electrode potential. I don't know if that answers your question, but we're getting, that's where we're moving
I'm
towards.
just wondering where you just kill it, where.
Well, you can definitely kill it. I mean, so you can go to very negative potentials and I guess generate, oxygen radicals and whatnot. But it's interesting you said that point about Mark Wrighton, because I'm very fascinated with turning on, as an electrochemist and a group of microbiologists, being able to turn on the cell, respiration and turn it off. And maybe you connected it for me back to my, my preschool days, playing in the Wrighton lab.
So from what I understand, the cells on the electrode surface move charge pretty efficiently, but the thicker you grow your biofilm, are there any significant loss?
Sure. So that's a great question. That's a great question. Is that a plant? Because this is my favorite slide, and I never get to talk about it much, but it really, I'm sorry. Okay. So this is an image of a cross-section of a biofilm schematic that's where I'm poising the potential at a positive potential so that the cells are dumping electrons onto the electrode surface. And the closer I get to the electrode, in terms of a hopping model, the more oxidized the cytochromes will be. But as I get further and further out, they're going to become more and more reduced. And so the portion that are oxidized drops from 1 to 0. And that's because cells are dumping electrons into them, and they're distributed throughout the biofilm, but they're only coming out at the edge, right? So if I'm a cell out here, I have two bottlenecks. First of all, there's a low relative abundance of oxidized cytochromes that can accept my electron. So I become essentially electron acceptor limited. The second thing is the gradient is shallower here. In other words, the difference in the oxidation state as a function of distance in the biofilm is shallower here. Once the electron is into a cytochrome, the rate at which it's going to move, if it's diffusion, is going to be proportional to this concentration gradient. It's fixed laws of diffusion, right? And so once it's in the cytochrome pool, it's going to be slower getting out. So I'm getting a double whammy. So the hopping mechanism predicts a finite biofilm thickness beyond which cells become electron transport rate limited and or oxidant limited. So that's the bottleneck, if I think you're asking about.
Here tomorrow, and have a chance to interact with this. So thank you once again for a great presentation.
Thanks.